Hello guys, I am Shubham from ShubhVed Tutorials. This video is about interrupted mating experiment. So before you start, subscribe to our channel and press the bell icon to get notified whenever the video is out. So let's get started with the video. So interrupted mating experiment is related to the conjugation. First of all, you have to know what is conjugation in detail. I have made the video. I will attach it in the I button. Do watch it because we will not understand what is interrupted mating if you do not know the conjugation concept. So this is related to HFR strains. Conjugation HFR into F minus mating. Right? This is the mating. And what is interrupted? Interrupted means this mating is interrupted. Okay, means stopped at various intervals. Okay, stop at various intervals of time. So this is interrupted mating experiment. Firstly, it was performed by, or we can say studied by Francis Jacob and Ely Oldman. So they were studying how the genes are transferred from one HFR strain to another HFR strain. One HFR strain to F minus strain, right? And what is the what we can say? How much time it takes? All we can understand, right? They used kitchen blender, right? Or we can say mixer, which we use in kitchen to break the conjugation pairs at various time intervals, and then which genes are traveled or entered first, then which is second, that we can understand with the help of interrupted mating. Okay, suppose we take here, this is a bacterial cell. Okay, and HFR, we know HFR is the F factor integrated inside a genome. Right, so this is the F factor, red one, and this is the double stranded DNA. Okay, chromosome or bacteria, blue. So this is, and the genes. Now we will draw the genes here with again a different color. So this is the one, STR resistant. Okay, streptomycin resistant. Then here, suppose I draw histidine plus. Then here, leucine plus and thiamine plus. And here, proline. Okay, and this is known as origin. We know origin. O. I will write here O. So now the HFR conjugation will start. So these are the two bacteria now. Conjugation bridge, right? Bridge-like structure. This is a donor. Okay. Here also we'll write donor. And here recipient. Recipient is having its own genome, right? Double stranded. Now what happened? HFR factor with first gene. First gene has come here. This is streptomycin R. Okay. Suppose I write here, this is after 5 minutes. Okay. Then the next gene. Now after 5 minutes, we break this pairs. So we'll find STRR gene here first, right? Then after 10 minutes, likewise after 10 minutes, we will see str plus next histidine plus, right? Then after 15 minutes, we will see str histidine, then leucine, like this. After every 5 minutes, we will see a new gene coming in, right? So this is interrupted meeting. We can understand the gene order. So this is what gene order. Which genes are coming first, then second, then third, and so on. Right. So first we can understand the gene order. Then second, time for gene, right, to travel. Here, for example, we saw 5 minutes are taken by each gene. Likewise, we can understand the time. And the third is the draw the map, draw the whole map. Means we can prepare a map, right? And E. coli genetic map was prepared from these experiments only, whole map of the E. coli genome. Whole map was prepared from this interrupted mating experiment. So let's see how the questions come related to this interrupted mating and conjugation. See, this is the first question. Interrupted mating experiments are performed using three different HFR strains. The three strains have different combination of selectable markers. Selectable marker means genes, right? The time of entry for markers for each strain is shown in the table below. So, see, here are the strains in the first column and the second time of entry, right? So, this is the first HFR strain 1. What we have to do using the above data, predict the correct sequence of markers means order of genes we have to understand on the E. coli chromosome. So see, methionine, threonine, streptomycin, phenylalanine, proline. Then one side of methionine there is threonine. Right now, here in the second, we will see what is common in the second. Methionine is common. Then histidine. Okay, means what we can write from this information. Methionine on one side is histidine and another side threonine. In the third HOS strain, methionine, see methionine, it is streptomycin and here it is histidine. So histidine is on the one side and another side here it is streptomycin. But before methionine and streptomycin, there is threonine here in the first strain, right? So we have written threonine. After threonine, it will be streptomycin, STR, right? And after phenylalanine is there, PHE, phenylalanine, right? Then phenylalanine, after phenylalanine, this proline, histidine, and proline is there in the HFR string. To the left of histidine is the proline, and here also histidine, proline, puromycin means phenylalanine, proline, and here it is streptomycin. After streptomycin, puromycin is there, and after puromycin, proline is there. Means between this phenylalanine and proline, there is puromycin. Okay, so what we'll write here? Puromycin and proline. So this is the order, right? See, now we will confirm. From the options, we can directly find out puromycin, then proline. Okay, see, puromycin is with proline, right? Then histidine, then methionine, right? Histidine, methionine, right? Then threonine. After methionine, threonine, streptomycin, and phenylalanine. All are correct. So this is the correct option. Then why other options are wrong? Let's see. You have to understand the sequence for this properly. See here, first option. Methionine is present with threonine, streptomycin, phenylalanine. Between phenylalanine and proline, there should be puromycin. Here it is given puromycin is afterwards and proline is in the between. So this sequence is wrong. Okay. The sequence should be phenylalanine, puromycin, and proline and histidine. Right. Rest other is correct. Here it should be the sequence phenylalanine, puromycin, proline. Same here is the second. See, streptomycin. After streptomycin, it should be phenylalanine. Right. It should be phenylalanine. So this is entirely wrong. See, streptomycin, phenylalanine. So here it is puromycin. Then here also histidine, methionine, phenylalanine. Wrong. Methionine, threonine is not there only. So this is completely wrong. Now here histidine, methionine. This is correct. Phenylalanine. Wrong. After methionine, it should be threonine. Then streptomycin. Then phenylalanine. So entirely the sequence is wrong. So B is the correct option here. So from the options we can directly see. Now the second question. Two HFR strains, right? Arginine plus leucine plus gal plus STR sensitive. HFR2 second strain. Arginine plus histidine plus galactose plus puromycin resistant and streptomycin sensitive. Were mated with F minus strain. F minus is arginine minus leucine minus galactose minus histidine minus puromycin sensitive and STR resistant. So the results of the interrupted mating experiment are shown as plots P and Q respectively. So these are the two plots shown. Okay, number of recombinants on the y-axis and time of interruption on the x-axis. Right. So we have to compare, see both the plots and then decide the order. Very easy. First arginine. We don't have to see the STR resistant because it is present in F minus. Right. So first is arginine. Here also, right? Then the second will be histidine. This is in the 10 minutes, right? Here after direct 20, it is leucine. So first arginine, then second histidine, correct? Then leucine. Fourth one will be galactose. And fifth one will be puromycin. So this is the order. Like this we can find. These are very easy questions actually. See, here it is arginine. First arginine is everywhere. But next here leucine. How leucine? After like arginine, there will be histidine. See, this is directly on 20 minutes. Before here, it is a histidine. Then here, same plots are there. 32 after 30 minutes, 32 might be galactose. And near 40, it will be 39, 38 something. Puromycin is there. So, like this, we can find the orders. These all our options are wrong. We can just see information is already given and we have to understand which genes are come first, what is the order, and thus see the options, find the answer, right? One more question we will see. The following table shows mapping data from 3 interrupted mating experiments using 3 different HFR strains and an F minus strain. Appearance of genes in F minus cells. See genes. Okay, see these are the genes that time HFR1, HFR2, HFR3. Genes and time are given. Following answers are derived. F and G, the distance and order. Okay, now let's see how we can understand the order. C, CDB. CDB, here also CDB. Here also it is CD. Okay, so CDB is common. Here also, here also. Okay, that's it. Then the next EF, EF, then FEG. Okay, EF means GEF, GEF, GEF. Okay, F and G means after C it is F, after F it is E. C, C, F, E, G. F after F it is E. So this is the correct option. P1. Q is wrong. Only slight difference is there. See carefully. I will write here Main difference is in that only. F, E, G. Okay. And in the Q option, it is written as E, F, G. So this is wrong. This is correct. From the strain 3, we can understand it. F, E, G. Right. So here is E. Between F and G is E. Here also. E, F, C are together. And after E, here it will be G. So this is order. P is correct option. So either A or either C will be correct. Now distance between, how can we find the distance between F and G is 32. Okay, F and G is 32 minutes. So just minus, F is 29 here, G is 61. Here also, F and G. Just minus, see, 63 minus 31 is 32. Right, so this is correct. F and B is 30. F and B, see, F is 24, B is 54. 30 comes, yes. As well as C and E is 28. C and E is 28. 47, 19. So yes, correct. B and C is 20. B, B and C. Here also B and C. Just minus 21 minus 1, 20. So R and S are also correct. So first one, correct combination is P, R, S. So much easy are those questions related to interrupted meeting. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever the new video is out. Also follow us on all our social media pages, and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.